welcome you back to this other session of ours, and today we are going to look at what we call mechanisms of photosynthesis. So today we are going to look at mechanisms of what? Photosynthesis. Now, actually here they are saying that photosynthesis is an oxidation reduction process in which water is oxidized to release oxygen and carbon dioxide is reduced to form what? To form carbohydrates. Now they are saying that photosynthesis is an oxidation reduction process. When we talk about oxidation process or oxidation reaction, so is a chemical reaction in which a molecule or an atom lose one or more electrons. And still oxidation reaction can be defined in terms of what? Loss of hydrogen atom. And still it can also be defined as the gain or addition of an oxygen atom. Then from there, when we shall look at what we call the reduction process or reduction reaction, and we are saying that is a chemical reaction in which a molecule or an atom gains one or more electrons. Still it can be a gain of hydrogen atom, or we can say a loss of oxygen atom. So we are saying that these mechanisms are divided into phases. So the phases of photosynthesis, one is the light stage. So if you don't call it light stage, you can call it the photochemical reaction or the Hill reaction. Then number two, we are having the dark stage or what we call the biochemical reaction. We shall look at each of these ones. Then from there, let us look at what we call the light dependent stage, and we say that the light dependent stage or the light dependent phase, this is a phase which takes place in the thylakoid membrane of the chloroplasts, as we are going to see. Now the main functions are, one is the photophosphorylation, then two is the formation of the NADPH. Now when we talk about photophosphorylation, we are saying is the addition of an inorganic phosphate to adenosine diphosphate, that is the ADP, to form adenosine triphosphate ATP using light energy. So that's the definition for photophosphorylation. Then we can also look at what we call the formation of the NADPH, which is the reduced form of nicotinamide adenine dinucleotide phosphate. Then without wasting a lot of time, let us look at the description of light stage of photosynthesis. Now we are saying that its reactions are triggered by light energy exciting photosystems 1 and 2 inside the thylakoid membrane at the same time, not one after the other. So these reactions occurs at the same time. And when we talk about photosystems, of course we have photosystem 1 and photosystem 2, as we are going to see, and we are going to look at how they operate. Now from there, not forgetting that photosystem 1 and photosystem 2, these ones run at different or they get excited at different wavelength or light of different wavelength of around 680 nanometers, that is for photosystem 2, and 700 nanometer, that is for photosystem 1 or PS1. Then from there, let us look at what we call the Z scheme, summarizing the light stages. Now when we talk about the Z scheme, somebody can say define the term Z scheme. Now Z scheme, it is a diagrammatic representation of electrons flow in cyclic phosphorylation and non-cyclic phosphorylation, showing the changes in energy potential of the electrons. Now when we talk about the Z scheme, this shows how the diagram which represents the flow of electrons in cyclic and the non-cyclic phosphorylation. Now when we talk about cyclic phosphorylation, yeah, let me show you it here. Cyclic phosphorylation, I'm going to show it to you. Yeah, this one is the cyclic phosphorylation. Now when we talk about cyclic phosphorylation, this is the process in which photosynthesis, where light energy is used to generate ATP from ADP and an inorganic phosphate without production of NADPH. Now we are saying that the cyclic phosphorylation is the process in photosynthesis where light energy is used to generate ATP from ADP and an inorganic phosphate without the production of the NADPH. It is also known as the cyclic electron flow. I'm talking about this part here, the so-called cyclic it is also known as the cyclic electron flow. Then it takes place in the thylakoid in the thylakoid membrane. Then from there we can go ahead and also look at the non-cyclic, because these two are sisters or they occur together. Now we talk about non-cyclic is a process in photosynthesis where light energy is oxidized to generate ATP and ATP and the NADPH from the ADP, an inorganic phosphate, and the NADP plus. So it also occurs in the thylakoid membrane of chloroplasts. It is also known as the non-cyclic electron flow. I hope that you have got the differences between cyclic and the non-cyclic. We said in cyclic there ATP is formed from ADP and an inorganic phosphate, but they don't form the NADPH. While in non-cyclic there we are seeing that the NADPH is being formed there. So from there, let us go and look at what we call the operation. Now let us look at operation of photosystem 1 and 2. Now we are saying that the chlorophyll molecule of photosystem 1, we are saying that the chlorophyll molecule of photosystems 2 and the photosystem 1 are excited by light of wavelength 680 nanometers and 700 nanometers respectively, causing the loss of electron to a chain of electron carriers in a series of reduction oxidation reaction as follows. I have already talked about the reduction oxidation reactions. Now from photosystem 1 OPS1, some electrons, as we are going to see, some electrons, now we are going to see that some electrons may flow cyclically. I talked about cyclic flow. We are saying to the ion complex up here, cyclically to the ion complex. Then from there it goes to what we call the cytochrome B6, which is here, the cytochrome B6. Then to what we call the plastoquinone, which is here, the plastoquinone is here. Then the cytochrome F, which is here. Then from the cytochrome F we can go to the plastocyanine, which is here, and finally back to the what? And finally back to P700, as you see there. And we are saying that during which electrons lose energy to form ATP from ADP and an inorganic phosphate. Then number 2, still in cyclic, we are saying that the non-cyclic, this one is the unidirectional, or we are saying that to the unknown molecule A. Then from there, the ion protein complex ferredoxin, yeah, what we are talking about is this side, the ion to the unknown molecule ferredoxin, this other part here, ferredoxin. Then to flavin adenine dinucleotide, what we call in the FAD, which becomes reduced to FADH, finally to ADP, then to form the reduced NADP, the NADP in bracket, what we are calling the NADPH. Then from there we can go ahead and look at then from photosystem 2, this one to the unknown molecule. From the photosystem 2 to the unknown molecule, there we are going to see from photosystem 2 to the unknown molecule, this one. Then from there you can go ahead and say from photosystem to the unknown molecule 2, then 2 is there, unknown molecule 2. Then from there we can say to the substance B, that is the cytochrome B. From cytochrome B to plastoquinone, which is P2, then the cytochrome F, then from there the plastocyanin, which is a copper enzyme, and finally to PS1, where there is 700. Now to replace the electrons earlier lost during this flow, electrons lose energy to phosphorylate ADP to ATP. There you are seeing it there, ADP to what? To ATP. Then the flow of electrons through carriers in the thyroid membrane releases the energy for active pumping of the hydrogen, from the stoma to the thyroid space. Then at the same time, photolysis of water occurs. Now when we talk about photolysis, is a chemical reaction that occurs when light energy is absorbed by a molecule, causing it to break down into simple molecules. And photolysis is divided into two. There is what we call homolytic photolysis and heterolytic. Homolytic involves the unpaired electrons, then heterolytic involves the paired electrons. So they are saying that this causes accumulation of hydrogen ions inside the thyroid space. 2 provides electron to replace those lost from photosystem 2 with the electron of oxygen molecule. Then we are looking at number 2 is the chemiosmosis. When we talk about chemiosmosis, this occurs, i.e. the highly concentrated hydrogen ion inside the thyroid space diffuses along the steep electrochemical gradient from the thyroid space via the stocked particle into the stoma, thereby providing 1 the energy to form ATP in presence of ATPase enzyme, then 2 hydrogen for reducing the NADP to form the NADPH, which together with the ATP form the enter the darker stage. Now from there we are saying that note the loss of 2 electrons by the chlorophyll in the photosystem breaches the chlorophyll molecule. In this state it can no longer absorb light energy effectively. Therefore the electrons lost to the electron transfer chain must be replaced. Therefore I sign out. We meet in the next episode or in the next video. Please like, subscribe and comment. By the way, in the comment section you can drop there your question. I'll be working upon them. I sign out. Recording.